Hello, College Biology. This is Nucleic Acids Part 4. It is still polymers, and we're going to talk about chromosomes. Chromosomes are really closely related to DNA because a chromosome is basically one long strand of DNA, and then it's associated with histones. So let's talk about that. DNA, as we are familiar with, is this double helix-shaped molecule, and they are very, very long, 200 or so, 100 to 200 nucleotides long, and that's a lot of string, if you will, in the nucleus. So there's got to be ways to manage that so that it doesn't get all tangled and jumbled and wild. And so when it needs to be packed up, it coils around these histones. So a chromosome is one long strand of DNA wrapped around histones. And histones are a protein that can be modified, and their shape can change a little bit. And when that happens, it causes the DNA to coil or uncoil. If you need to use an area of DNA, that area is uncoiled, and if the cell doesn't need that area of DNA, it could be coiled. So, and when a cell is undergoing cell division, the DNA has to be condensed down so that it can be easily moved, so it needs to be coiled. And so that coiling and uncoiling process is facilitated by histones, which the DNA wrap around when it needs to. So that's a chromosome. It's a strand of DNA wrapped around histones. Now, in human cells, there are 46 strands of DNA. These are 23 pair. They come one from each parent. And so this chromosome here and this chromosome here have instructions for very, very similar jobs. And one came from mom and one came from dad. And we can count out these 46 chromosomes. This number changes from species to species. So different organisms will have different numbers of chromosomes. For instance, fruit fly have 8, pineapples have 50. We've got 46. And all of our cells have the same 46 chromosomes, and on those chromosomes are our 20,000 genes. So those chromosomes are divided into two regions, or at least there's two conceptual regions associated with chromosomes. These are non-coding and coding. So what does that mean? Well, our coding regions are all the genes that we use. We copy them into RNA so that we can make protein. So all of these genes are our coding regions, and that composes 2% of our chromosomal DNA. Only 2% of our DNA is actually coding for genes, which means 98% of our DNA are non-coding regions, and these are the spaces in between the genes, as illustrated here. So that's kind of interesting. So what's going on with those non-coding regions? We know the coding regions, 2%, that's all of our genes that are going to be copied into protein. But what about that other stuff? Well, there's some ideas about how all that non-coding regions came to be. One, DNA can be mutated, and that means there can be a change in the sequence. DNA can be altered. The nitrogenous bases can change. And the most common form of radiation on Earth is the most common source of mutations on Earth are radiation. So if you have something like UV radiation coming in, it can transfer energy to those nucleotides and cause them to make a bond with each other they wouldn't normally make, or change to a different nucleotide, or remove a nucleotide, or add a nucleotide. All of these are possibilities. And so when you have a mutation, you've changed that DNA. And when you change that DNA, lots of things can happen. One of the things that can happen is that gene is no longer readable. And if you can't read it and make RNA, then you're not going to make protein, which means this gene, if this gene gets mutated a specific way by radiation, it will no longer be made, which means it will become a non-coding region. So you can deactivate genes with radiation, and you can turn a coding region into non-coding region. As that builds up over time, you can generate more non-coding region. There is another suggestion as to where non-coding regions come from, and that are viruses. There are types of viruses that drill into the cell, release their nucleic acids, then find a way to get their nucleic acids into the nucleus, where they will insert themselves into the chromosomes of their host. So there can be dormant viral DNA, and maybe it's been deactivated through mutation too, that has inserted itself into our chromosomes. And those can build up over time. And so if it happens to me, and so I have extra DNA in my chromosomes, and then I pass that on to my children, then they have it, and it can go on and on. So generation after generation, we can collect these small snippets of DNA that slowly add up into our chromosome to contribute to these non-coding regions. So, quick review. Nucleic acid, this is our last polymer. A chromosome is one long strand of DNA wrapped around histones. We have 46 or 23 pair per human cell. Different species have different numbers, and all the cells in our body have the same chromosomes. 2% of those chromosomes are coding regions, which contain all of our readable genes. And 98% are non-coding regions, which is the space in between the genes that are not read to be made into protein. They are, those sources of those can include genes that have been deactivated by mutation and ancient viral deposits that have actually inserted new DNA into our chromosomes. I hope this helps. Struggle well.